This is the screencast for Monday, April 21st, lecture on Module 5, Lecture 3. A couple brief announcements before we get through it here. Your exam 5A is next Monday, April 28th. That will be on carbohydrates and amino acids. And you'll have to refer to a website for some of those questions on that test. Exam 5B is next Wednesday, April 30th. That will be on proteins and DNA. And you'll have to refer to a couple literature sources that will be linked through Canvas to answer those questions. In this lecture, we're going to go over amino acid structure, properties, and sequencing of peptide chains. So we'll start with amino acid structure. We'll look at a couple different representations of them. So there's 20 proteinogenic, naturally occurring alpha amino acids. And again, we're using the term alpha to denote the carbon that's next to a carbonyl. So you can see that in the structure here. On the alpha carbon is the amine group. So notice that that is a primary amine. There is one case where there's a secondary amine. And notice that the naturally occurring alpha amino acids have the S configuration. So again, that's the Kohn-Ingold-Prelog designation. And when we look at that from a Fisher projection, where your eye is placed right here, and you convert that, naturally occurring amino acids have the L designation. So that means the amine group is on the left of the Fisher projection. The acid at oxidation state 3 is on the top. And then R1, the side chain, is on the bottom of the Fisher projection. So the common structural features with all of these amino acids, I'll highlight that in green. So you have the acid, you have the alpha carbon, you have your hydrogen to the right, your amino group to the left. So that's the common features. And then what differs is this side chain R1. So the nature of that side chain R1 can be classified into 4 different groups. So we have nonpolar groups. Those include glycine, alanine, valine, leucine, isoleucine, methionine, proline, phenylalanine, and tryptophan. We have polar side chains. Those are asparagine, glutamine, serine, threonine, tyrosine, and cysteine. Acidic side chains. Those include aspartic acid and glutamic acid. And then finally, the basic amino acids. Those include arginine, histidine, and lysine. So really, it's the nature of the side chain that makes them different. So we'll go through those 4 different groups, look at what makes them fall into those different categories, and we'll introduce a little bit of the nomenclature now. So the most basic amino acid is glycine. And so you'll note for each amino acid, there's the actual name. Then there's a 3-letter abbreviation. So glycine is GLY, gly, and it's further abbreviated as G. Glycine is the only one that does not have a side chain. Essentially, the alpha carbon is 2 hydrogens. And so moving on now to L-alanine. Remember that the L refers to having the amine group on the left side of the Fisher projection, and that corresponds to the S stereochemical designation. So L-alanine, abbreviated ALA or A, that has a methyl group, so that's the side chain. So the side chain highlighted in red there, that's a methyl group, CH3. So what you'll notice about all of these nonpolar amino acids is basically they're very carbon rich. And what you see moving along, they're pretty short looking side chains. So valine has an isopropyl group. So that side chain is isopropyl. Leucine is isobutyl. Isoleucine, notice that has an additional stereocenter at the beta carbon, so I'll highlight that with an asterisk. So that's a secondary butyl. Methionine has a thioether side chain. Proline is the only one that's a secondary amine. So notice that the side chain wraps back around to form a ring with the alpha carbon. So this is a secondary amine. Phenylalanine has a benzyl group. And tryptophan has an indole. So as you advance on to biochem or other cellular and molecular biology classes, you'll want to know the structures as well as the 3-letter and 1-letter abbreviations of these. Moving on to the polar amino acids. Asparagine has a primary amide as the side chain. Glutamine is also a primary amide, but what you'll notice is there's one more methylene group. Serine has a primary alcohol. Threonine, another stereocenter here. This is a secondary alcohol. Tyrosine has a phenol, so that was the hydroxybenzene. And then cysteine has the thiol. The acidic amino acids have a second carboxylic acid. So aspartic acid. And then glutamic acid, you'll notice the extra carbon linker in there. The basic amino acids, arginine at the end of the chain has this functional group called a guanidine. Histidine has this heterocycle called a imidazole. So we talked about imidazole in terms of nitrogen basicity. You have two different sp2 type nitrogens. And remember, like, where the location of the lone pair is, that determines the basicity of that lone pair. And then lysine has another primary amine at the end of the chain. So an alpha amino acid has both the acidic carboxylic acid functional group and the amine, which is basic. So when they're shown in this representation, notice that everything's neutral. However, we need to consider the response of the amino acid in the carboxylic acid group at different pHs. And so the reason for that is we're going to consider these molecules now under lab conditions from pH 0 to 14, but then under physiological conditions around 7. And so we want to have an idea of what sort of charge species exist for those structures. And then as we start making amide or peptide bonds, what's the overall charge of the peptide chain at different pHs? And so that's determined on both the acidic and basic functional groups, but also the side chain as well. So let's recall that the pKa of an acid is about 5. And now the pKa of a protonated amine is about 10. So when we say that, we're saying the protonated amine, that pKa is about 10. So when that gets deprotonated, it just becomes the neutral amine. So this is called the ammonium, typically a salt, so there's some counter ion, and then that's the amine. So the exercise that we're going to do is look at how the charge of the amino acid changes as we adjust the pH from 0 to 14. And something we want to keep in mind is this calibration point of pH 7, which is about physiological pH. So remember that on the pKa scale, when you're between 0 and 14, pKa is equal to the pH. So due to the different structural considerations, the pKa has, the acid pKa is around 2, and the ammonium is around 9. So remember that this is the protonated amine. So we can determine what the charge of the alpha amino acid will have at what pH based on this information. And so the reason that's important is when we're making increasingly long peptide chains, and remember a peptide is just a polymer of amino acids. One way to purify them is using gel electrophoresis. And so that's based on the mobility of that chain on a medium where an electric field has been applied. So due to the nature of the R1 side chain, alpha amino acids will vary in their charges as the pH scale is spanned from 0 to 14. So essentially this is like a titration, an acid-base titration. Most typically the charge will vary from +1 to 0 to -1. So you can make the association of acidic to neutral to basic when we're going through this acid-base titration curve. So when you're neutral, there's a special name given to the structure of the amino acid, and that's called a zwitterion. And a zwitterion is basically an internal salt. So you have a plus charge somewhere, and then you also have a negative charge somewhere in the same molecule. That's called a Zwitter ion. So as an example to start, we'll look at alanine. So remember the structure of alanine. As the methyl side chain. And let's just start at pH of 0. So at pH 0, we know that on the pH scale we're under acidic conditions. So take, for example, we have alanine, we've added, say, HCl to it. That's going to result in the amine being protonated. So when the amine's protonated, it forms the ammonium. And again, if we were using HCl, this would be balanced off by a chloride anion. So now when we look at those functional groups, we have the ammonium. We look at the overall structure, we see there's a positive charge. So we would say under those conditions, at pH 0, the structure has a charge of +1. The pKa of the acid is 2.34. So if we're moving in this direction, we hit this point. So now we're adding in, say, base. We hit 2.34. The acid gets deprotonated. And so when that happens, you end up with this carboxylate. And then the amine is still protonated. So that's still the ammonium. And now you're at this point where it's the internal salt or Zwitter ion. So now you have a plus charge, a minus charge, and overall for the structure, the charge is 0. Now, we keep dribbling in base in our titration, and eventually we're going to hit this second pKa2, that should read pKa2, at 9.69. The ammonium group then gets deprotonated to the amine. We lose that plus charge, we're left with the negative charge. And now above 9.69 to 14, overall that amino acid has a -1 charge. So we can do this for all of the 20 proteinogenic amino acids, and really what changes is sort of the number for the pK1 and pKa2, and then depending on the side chain, if it's acidic or basic, we can have another pKa value in there. So the consequence of these different ionic forms of alpha amino acids is something called the isoelectric point, or Pi, and that's the concentration at which the Zwitter ion is greatest. So remember, the Zwitter ion is that neutral form where overall the structure has no excess of charge, so it's neutral. So the isoelectric point, Pi, is calculated as pKa1 plus pKa2 divided by 2. So in the case of alanine, which we just looked at, the Pi value is 6.02. So meaning that at pH 6.02, the primary form of alanine is the Zwitter ion, and that has no charge. So this is the Zwitter ionic form of alanine. So notice the plus charge, the minus charge, overall that's neutral, that's the Zwitter ion. The concentration of that species is greatest at 6.02. the bigger picture of this is that by adjusting the pH of the medium, we can change the charge of the amino acid. And again, that's going to help us in terms of purification of these things. If we have mixtures of different, say, peptide chains, they'll have different mobility on the gel electrophoresis solid support material. So generally speaking, the amino acids can be grouped together to get an idea of their ionic forms at different pH values. So this table has the pI values for the nonpolar amino acids. So again, those with the nonpolar side chains. What you see are the different amino acids. The pKa1, that relates to the carboxylic acid. The pKa2, that relates to the ammonium NH3. So you take those two numbers, add them together, divide by 2, and then you get the pI value. And what you find in general for these nonpolar amino acids, the pI is approximately around 6. You'll notice that there's some variation in these numbers, and again, that depends on what the nature of the side chain is. So on these LEAP questions, you're presented with different amino acids. You have the pKa values. So for proline, again, that's the secondary amine. pKa1 is 1.99, pKa2 is 10.6, and the pI is 6.30. What is the form of proline at pH 13? So to solve these, what I like to do is just draw the pH scale, put in these different pKa values, put in the pI value, and then determine what pH the system is at. So we'll start off with just drawing a line. So 0, that's the acidic pH. 14, that's the basic pH. The midpoint 7, that's the neutral or physiological pH. Let's go ahead and plunk in our pKa values. So 1.99, that was pKa1. 6.30, that was our pI value. That's the Zwitter ion concentration. So 10.60, that was pKa2. And it says we're operating at pH 13. So what is the form of proline at pH 13? So we know that we're well past the pK of the carboxylic acid being deprotonated. So from that, we can eliminate A. That has to be deprotonated. We're well past the pK of the ammonium group being deprotonated. So that has to eliminate C. And basically, we need to end up now with the neutral amine. So B is going to be the correct response. D is not correct because the pKa value of, say, just a regular NH2 group to form the negative charge on the amine, that pKa is around 35. So that's well past the pH scale. And remember, we're only operating between 0 and 14, where pH is equal to pKa. So that ammonium has to become the amine. So we're looking at this pKa2 value where ammonium gets deprotonated to the neutral amine. That's how we arrive at B. So at pH 13, this is going to be the structure of proline. So the rest of these questions I'll let you do on your own so that you can draw that scale out, stick the pKa values in, stick the pI value in, look at what pH you're prompted to consider, and then go through the choices. So again, here's the pI values for the polar amino acids. So these were asparagine, glutamine, serine, and threonine. Notice there's still the two pKa values, 1 and 2. The pI values, we can say, are approximately, say, 5.5, give or take. And again, that depends on the side chain. When we move to the polar amino acids with the alcohol or thio groups, so say cysteine or tyrosine, what you'll notice now is there's an intermediate or a terminal pKa value. So now we have 3. pKa1, that's always the acid. pKa2, this is where things will change. So for cysteine, that's the thiol group. And then for pKa3, that was your regular ammonium. For tyrosine, you have your pKa1, that was the acid. Your pKa2 was the ammonium, and now this new pKa3, that deals with the phenol of tyrosine. Notice that these are from 5 to 5.5, and again, the side chain, either the thiol or the phenol, will lower that pI value. In this LEAP question, you're asked to consider now tyrosine. So again, that has the acid pKa value, the ammonium pKa value, and now this third pKa value due to the phenol. The pI value is at 5.56, and you're asked to consider what is the structural form at pH 11. So go ahead and draw out that scale, put the 3 pKa values in, the pI value, and then look at the pH of 11. You'll do the same thing for tyrosine, but now we're lowering the pH to 9.5. So now you're basically going from basic pH, you're dribbling in acid in the acid-base titration, you're headed towards 0. So adjusting the pH of tyrosine even more by adding acid, you're at pH 4. Look through those choices and determine what functional groups will be charged. And then finally, we're ending up at acidic pH 1. So of these four questions, you're looking at choices A, B, C, or D to determine what groups are protonated or deprotonated depending on the pH. For the acidic amino acids, there were 2 of them. And now these will necessarily have to have 3 pKa values. So pKa1, the carboxylic acid of the carbonyl that's bonded to the alpha carbon. Your pKa2, that's the acid of the side chain. And then your pKa3 of the ammonium. And then overall, we can say the average pi value is around 3. So by adding that second carboxylic acid group, that drops the pi value quite a bit towards the acidic region of the pH scale. So you can run through these LEAP questions now with aspartic acid in consideration of now the 3 pKa values. Notice the pi value is 2.77, and then for this question you're considering the pH of 2.5. So basic amino acids. These as well have 3 pKa values. So pKa1 is your carboxylic acid, pKa2 for amine group 1, and then pKa3 for amine group 2. Notice the pi values are shifted above the neutral value of pH 7, and then they'll vary based on what the nature of that side chain is. So remember, for arginine you had a guanidine side chain, histidine you had the imidazole ring, and then for lysine you had another amine. This question, subsequent questions look at histidine. Again, the side chain is the imidazole ring. So you have your pKa1 at 1.82, pKa2 at 6.0, and then pKa3 at 9.17. The pi value is just above pH 7. And then you're looking for the form of histidine at pH 7. So draw up the scale, put in 0, 7, 14, add your pKa values, then consider what pH the medium is at, look at the possible responses in terms of what functional groups are going to be deprotonated still, what functional groups are going to be deprotonated. So overall, that exercise, you were looking at individual amino acids, but you can imagine once we start bonding them together through amide bonds, which are also called a peptide bond, ultimately a long peptide chain will make a protein. The purification of those things is done using gel electrophoresis. And the method employs a solid phase gel in which a pH buffered medium has an applied electric field to it, and the molecule will move through that gel according to the pH value and then the electric field that's applied. And really the pi value is important because you can make things migrate based on the overall charge of the molecule. So due to the fact that the medium is buffered, again, that's the pH value, the two situations can exist. If the pi value is greater than the pH of the buffer, the molecule will be overall positively charged and migrate towards the cathode, the negative electrode. If the pi is less than the pH of the buffer, the molecule will be overall negatively charged and migrate towards the anode, the positive electrode. So these migrations are basically separations if you're trying to purify a mixture of things. So in essence, it's like thin layer chromatography, but perhaps a little bit more complicated. So now that we know the structures of the amino acids, we know how to analyze their protonation states at different pHs. We can figure out that charge. The next thing we need to look at is the peptide sequence convention. So alpha amino acids are monomers that are used in peptide synthesis. So a peptide can be considered a biopolymer. So basically, a polymer is just a sequence of monomers bonded together. What's interesting about these biopolymers is that the monomers themselves do not have to have a repeating symmetry. So in sort of traditional polymer synthesis, you have, say, one or two monomers you're bonding together, and you'll get this sort of repeating unit. However, in these biopolymers, you don't have to have that repeating unit in terms of symmetry. Overall, you just look at, there's these 20 different proteinogenic amino acids. Those are the monomers. Amino acids are bonded together through the amide bond. In the context of these biomolecules, we call them peptide bonds. So how do we spot that? So we're looking for the oxidation state 3 carbonyl, and so that is an amide bond. So alternatively called a peptide bond. So in terms of analyzing the sequence convention, so now we're taking the individual amino acids, bonding them together to get a sequence. How do we read that sequence? So we're going to read it from left to right. On the left side is called the N terminus. On the right side is called the C terminus. So the structure that's shown on the left, when we look at the side chains, we see that this side chain highlighted in red is a methyl group. We go over to the next amino acid. We see that this side chain highlighted in blue, that's an isopropyl group. And we would say that that's alanine bonded to valine through a peptide bond. So we would call that alaval or AV. Notice now in the sequence on the structure on the right-hand side, we have valine on the left side, alanine on the right side, and so that's now a different sequence, ala or VA. That's an important difference to realize, because ultimately that sequence will give the peptide chain its structural features and ultimately its function. So when you get a large enough peptide string together, these things make up proteins. Another word for a protein is an enzyme. And so enzymes are basically biomolecules that carry out chemical reactions under physiological conditions. So in this class, we're focused really on the organic functional groups, the structural features, but you have to imagine that you can have these sequences being very long, and ultimately they'll have other structural features associated with them that imparts their chemical reactivity. So there are 4 elements to those protein structures. So we're talking about imagining very large biopolymers here. The primary structure is the exact sequence of amino acids as we read from the N terminus on the left to the C terminus on the right. The secondary structure, that's due to all the non-covalent interactions present. So typically, we're most familiar with hydrogen bonding. So I'll abbreviate non-covalent interactions as NCI. There's other non-covalent interactions that we'll talk briefly about, but hydrogen bonding is probably the most prevalent. The tertiary structure is the complete 3-dimensional conformation. So based on the primary, secondary structures, how does that give rise to the overall conformation? What does it look like? There are different shapes that can occur. And quaternary structure is the association of 2 or more peptide chains. So again, as we're reading this, we read from left to right, so from the N to the C terminus. N to the C terminus. So if we're given a biopolymer that consists of many amino acid monomers, how do we determine primary structure? So that's the exact sequence of those amino acids. So we'll go through a couple different chemical and enzymatic methods to determine what that sequence is, reading it from left to right. So one very general chemical reaction is amide hydrolysis. So that requires forcing conditions with a strong acid, such as 6-normal hydrochloric acid, heated to about 100 degrees, so that's refluxing water. And what that's going to do is hydrolyze this bond. And give us the 2 amino acid monomers. So when we do that with this AV dipeptide, we can calculate that has an exact mass of 188.11609. That in and of itself is a way to determine what amino acids are present, but it doesn't really tell you the sequence. So when we hydrolyze it, we'll find that we end up with alanine and then valine. And then say, for example, we were able to separate those 2 things, run mass spec on them. We would find that alanine has that mass, valine has that mass. And then we could say, yes, that dipeptide consisted of those 2 amino acids. However, we still don't know the primary structure. We've only identified the constituents. We don't know their sequence. So instrumentation such as NMR and mass spec can tell you that those amino acids are present, but they don't tell you the sequence. So what other methods can we use to do that? So here are some LEAP questions that you can go ahead and answer to practice all the concepts that we've learned so far. So given AV, how many unique C13 signals would that dipeptide have? How many of those signals would lie in the 180 to 220 region? So remember that on the NMR spectrum from 220 down to 0? Where do the different functional groups lie? How does hybridization affect the carbon chemical shift? So here's a question. Digestion of an unknown peptide sequence shows 3 amino acids present by NMR and mass spec. How many possible sequences can there be? So what you can do to figure this out, just consider, say you have 3 amino acids, say glycine, alanine, valine. How many other ways can you write that sequence? So just rearrange those letters so that you get unique, distinct sequences. And how many possible ways can you do that? So see if you can figure that out. So in general, the possible combinations for an amino acid sequence, if no amino acids repeat, the number of possible sequence combinations is n factorial. If amino acids do repeat, the number of possible sequence combinations is n to the m. So n is your number of amino acids present, and m is the length of the peptide chain. So you can plug those numbers in there, help you figure out what are the number of distinct, unique sequences you can get from n amino acids. Those fall again into 2 categories: if none of the amino acids repeat, or if you do have multiple of the same amino acid. So again, we're trying to figure out what is the sequence. So we'll run through some chemical methods of doing that, and then some enzymatic methods. Some of the first chemical methods that were developed sequence the chain through successive cleavages, starting from the N terminal. So that's the left side of the sequence. So you have this long sequence, you're starting at the left side, and you're chopping off that N-terminal residue, 1 amino acid at a time. The reason for that is because you have this group here that's shown as the ammonium. As you adjust the pH, you can get to the amine group, and basically you run a reaction on that amine. So this is an example of the Edman degradation. So the Edman degradation uses this reagent called phenyl isothiocyanate. So here's the phenyl ring, and then these 3 atoms here and that bonding pattern are called an isothiocyanate. What's important to note is the central carbon, which is sp hybridized, has an oxidation state of 4. So we've seen throughout this semester that carbon at oxidation state 4 is electrophilic. It wants to undergo an addition reaction. That will either keep that oxidation state at 4 or reduce it down to 3. In this specific reaction, the oxidation state is conserved because you're adding that amine group to that carbon. So the amine of that N-terminal side chain adds to it. And then you get a cyclization event that forms this new heterocycle called a hydantoin. So phenyl thiohydantoin. And we can see the elements of that isothiocyanate now bonded to that N-terminal residue. So it specifically cleaves there, and it's tagging that N-terminal residue. And the consequence of that, with that tag highlighted in red there, you get a very distinct exact mass for each unique amino acid. So imagine that you have the 20 proteinogenic amino acids. Now imagine they're in that phenylthiohydantoin form. They're all going to have unique masses. So when you separate out that mixture from this Edmond degradation, you can see what the mass of that hydantoin is, and you can say, okay, because that one is tagged, it had to be on the N-terminal. And then you're left with the mass of the remaining chain, in this case from the dipeptide that's valine. You find its exact mass, and you can say, okay, the other amino acid bonded to it was then valine. So that's going to give you the exact sequence of AV. So as a practice, if you consider the tetrapeptide cis, ser, val, phi, if that was subjected to 2 consecutive cycles now of the Edmond degradation, what peptide would remain? So again, this is the N terminus, that's the C terminus. The Edmond degradation works by moving from the N-terminal residue towards the C-terminal residue. If you apply that Edmond reagent twice, what peptide remains? Another reagent that's used to carry out N-terminal residue analysis is called the Sanger reagent. The Sanger reagent is 1,4,2,4-dinitrobenzene. This does the same thing conceptually as the Edmond degradation. It's going to utilize the reactivity of that N-terminal amino group, so N-terminal, C-terminal, and it's going to tag that group with a molecule that has an exact mass. So what happens is this fluorine gets substituted. So now you're forming this bond. So the fluorine has been displaced, it's substituted by that N-terminal group. Now you have a tag on that, and you can basically undergo a hydrolysis reaction that will cleave that bond, and you'll see which amino acid is tagged. And so again, the same thing, you can have a library of these Sanger tagged amino acids, look at their exact masses, and then find out, okay, since that one has the tag, it had to be at the N-terminal. So Sanger, for his work in this area, actually won 2 Nobel Prizes. That's very impressive. For his investigations on the determination of the structure of proteins. So if you want to cleave off and find out exactly which amino acid was tagged, you have to do that hydrolysis. So you can do that under basic conditions. Again, we're going to cleave that bond. We find out that the alanine was tagged and when it's tagged with that reagent, it has that exact mass. So from the mixture, we separate, we find out what the masses of the different products are. We look at our library and we say, okay, this one matches alanine when it's tagged with that reagent. So we know, therefore, that alanine had to be at the N-terminal residue. So again, one of the issues with this is you're chopping the sequence up one residue at a time, so it's very lengthy. And you can imagine if this side chain goes longer and longer, when you get to this hydrolysis step, you're really only able to identify what one of the N-terminal residue is. You don't have any other sequence information moving down the chain. So how can we use some more chemical reactions that will help us determine this without doing a whole lot of work? So as we move forward in determining primary structure, again, that's the exact sequence of the order of the amino acids. Remember, there was 4 types: polar. So in these slides moving forward, this sort of light bluish color will be our polar amino acids. Our non-polar will be these sort of light red or pink. The acidic amino acids will be the dark red, and then the basics will be the dark blue. So within each of those circles are the 3-letter abbreviations, and we're going to learn some methods of how we can cleave a long chain into defined segments that will help us analyze the sequence better. So here's some logic that we can apply to understand the difficulty in what we're trying to achieve here. So it says a peptide with 12 amino acids has the composition, so composition, not the sequence, of these amino acids. So those are all the amino acids present in that peptide, but that's not the sequence in which they're bonded. Furthermore, it says that cysteine, cis is the C terminus, Sur is the N terminus. We take that 12 amino acid chain, subject it to partial acidic hydrolysis, and we end up with 7 sequences shown. Then the series of questions ask you to identify the different amino acids in the sequence, and then overall, what is the exact sequence? So the first question here is, what are the first 3 amino acids of the sequence? So looking at that, using the information known, again, operating from the N to the C terminus, you want to put these fragments together so that you get the exact sequence. So what are the first 3 amino acids? What are the second 3 amino acids? What are the third 3 amino acids? What are the last 3 amino acids? So at that point, when you do that, you should have overall the exact sequence from the N to C terminus of the 12 amino acids present. So to take that problem a little bit further, once you have your sequence of the 12 amino acids, you're going to navigate to this website, Baychem. And so they have a couple different things in which you can calculate properties based on your amino acid sequence. So the first question is, what is the molecular weight of the peptide? You're going to enter the amino acid sequence either in the 3-letter abbreviation or the 1-letter abbreviation. Pick the appropriate nature of the N terminus and the C terminus, so those will occur in little drop-down boxes you'll select. And then what's the value of the molecular weight? What does it calculate to be? So as I mentioned in the beginning of this screencast, this will be one of the websites you'll have to access for your exam 5A that you'll take next Monday to determine some properties of these peptide chains that we're going to look at. So the website, when you put in your sequence, will also calculate your isoelectric point. So now it's looking at the individual amino acids. It's going to calculate that PI value for you. Overall, what is the net charge at pH 7? So all of this stuff is calculated. It's put out on a printout. Once you enter your sequence and you choose the appropriate N terminus and C terminus functional groups. What is the average hydrophobicity? So hydro means water, phobicity means hating. And so, again, depending on the nature of the side chains, once you get your sequence of the amino acids, then what happens is these things can start hydrogen bonding within the molecule itself. It'll take on a certain shape, and then you'll have side chains sort of pointing outside of the molecule, some inside. And these terms, hydrophobicity, imagine you're, under physiological conditions. So you're in this aqueous environment. You want your polar groups sort of pointing out towards the water, your non-polar groups pointing inside. The nature of those individual amino acids will determine that in the secondary structure. And then what is the ratio of the hydrophilic residues to the total number of residues? So hydro, again, is water. Philic means loving. So how many water-loving residues to the total number of residues? And as you look at structures from the organic perspective, the side chains are what are going to determine these properties ultimately. So again, that website is what you're going to navigate to for your exam 5A to answer some of the questions that you'll see on that exam. Things like molecular weights, PI values, hydrophobicity, hydrophilicity, PI, to calculate those values based on some questions you'll see on exam 5A. So aside from partial or total hydrolysis with HCl, again, those are not specific. So once you do that, you just end up with this collection of amino acids. You could separate them, identify them using instrumental methods, but again, it's not telling you the exact sequence. There's a reagent called cyanogen bromide. that's going to cleave a peptide chain in a very specific way. So cyanogen bromide acts upon methionine residues only. So here we have this chain, reading again from the N to the C terminus. That's the sequence. And then you can see the different colors representing the four classes: nonpolar, polar, acidic, and basic. When you treat that peptide with cyanogen bromide, you say you're reading left to right. When it hits a methionine residue, what it's going to do is get past it and then cleave that chain. So it's going to cleave the chain right there. And when it does that, you now end up with two peptides. So this one with 6 amino acids. So we could call that an oligopeptide. And then 2 amino acids, we would call that a dipeptide. So now look what's happened. We have our N terminus, and now we have methionine at the C terminus of one of those chains. Then the dipeptide, we have the N to the C terminus. So the important thing to note is that if you do have the methionine amino acid in there, the cyanogen bromide will cleave it, and then methionine will end up as the new C terminal of one of those peptides. So again, if we want to know the exact sequence of a protein, otherwise called an enzyme, which are large biomolecules that catalyze chemical reactions under physiological pH, we need some more methods to help determine the exact sequence so that it's not as time intensive. So there are these enzymatic methods. So we can use enzymes to help us cleave peptides in a very specific way. The ones that we're going to learn about are called serine proteases. So enzymes that end in aces, they do cleavage reactions. And these serine proteases will cleave specific enzymes, specific amino acid residues in a very selective fashion. So these serine proteases are also called endopeptidases. So there's 3 of them that we'll look at. The first one is called trypsin. So trypsin cleaves a peptide chain at the carboxyl group of basic amino acids. Those include lysine and arginine. So overall, what does that mean? Trypsin will kind of read the chain from the N to C terminus. When it hits either lysine or arginine, it's going to read it and then cleave. So lysine or arginine will end up at the C terminus of that new fragment. Chemotrypsin cleaves the carboxyl end of aromatic amino acids, including phenylalanine, tyrosine, and tryptophan. So again, as it's reading the chain from the N on the left to the C on the right, if it encounters phe, tyr, or tryp, it's going to read it, it hits it, and then cleaves it. So those amino acids will end up at the C terminus of the newly resulted fragment. And then, as opposed to the Sanger and Edmond degradation, the carboxypeptidase cleaves amino acids starting from the C terminal residue. So this actually reads right to left. It will sequentially cleave off every amino acid at the C terminal one by one. And so if you can control the rate at which that's done, it's giving you information from the C terminal residue. So again, trypsin cleaves a peptide chain at the carboxyl group of the basic amino acid lysine and arginine. So let's look at 2 examples of this with trypsin. So how many amino acids we have? 1, 2, 3, 4, 5, 6, 7, 8. We have 8 amino acids in this first chain, so we would say this is an oligopeptide. So we're reading from N to C, and we're looking for lysine or arginine. And so those were the ones colored in dark blue. So as we read, proline, glue, lu, lyse, we hit a lyse, so it's going to cleave there. And then we end up with 2 fragments, each with 4 amino acids. And notice now the lysine is at the C terminal of one of them. In a second example, we read from N to C. We're scanning over 1, 2, 3, 4, 5, 6. We hit an arginine. We're going to cleave there. We end up with these 2 fragments now from N to C. One of them has 6 amino acids. The other one has 2 amino acids. And the arginine ends up at the C terminal of that longer chain. So really this is taking something very long, it's chopping it into these defined fragments based on the action of the endopeptidase. Chemotrypsin cleaves a peptide chain of the aromatic amino acid residues phenylalanine, tyrosine, and tryptophan. We have 3 examples here. So 1, 2, 3, 4, 5, 6, 7, 8. So all of these have 8 amino acids in their sequence from N to C. So as we're scanning along, we're looking for phi, tyr, or trip. We start in this first example. We get to the second residue, that's phenylalanine. It's going to clip it there. So it clips it. Now we end up with 2 amino acids. Phenylalanine is at the C residue, C terminal residue. Then we end up with this 6 amino acid fragment. And again, we can identify those either through, say, mass spec methods or NMR. The next example, as we scanned through, we see we have tyrosine, so it's going to clip there. We end up with a 5 amino acid chain, tyrosine at the C terminal, then a 3 amino acid chain, and then the last example is using tryptophan as the amino acid that's going to get cleaved. So Again, we're scanning along from left to right. We hit that tryptophan. We then cleave. That now ends up at that C-terminal residue of this 5 amino acid chain. So these enzymatic methods, again, are going to scan for specific amino acids, either the basic or the aromatic, and cleave them in a selective manner. The last one is carboxypeptidase, and that's going to clip off the C-terminal residue. So from N to C, when we treat with carboxypeptidase, it's scanning along till it gets to the end, and then it's going to clip there. So we go from an 8 amino acid residue now down to 7. And then we see that glycine was produced. If we let this go long enough, that carboxypeptidase will now cleave off from that 7 amino acid sequence proline, and we'll end up with a 6 amino acid sequence. So proline would then start showing up in the reaction medium. The 6 amino acid sequence, again, the reaction keeps going. Carboxypeptidase comes along, it's reading it, it sees the arginine, it cleaves. We're now down to 5. And arginine starts showing up. So again, so first to show up, second to show up, third to show up, so on and so forth, as time progresses. One more complicating factor about primary structure is the presence of disulfide bonds. So that is a disulfide. So 2 sulfur atoms bonded by a sigma bond. And those result from cysteine residues. So 2 cysteine residues basically being oxidized to form the disulfide bond. So in the periodic table, CNOF. So sulfur is below oxygen. It's prone to undergoing these type of reactions to form these disulfide bonds. And so you can imagine you have this long chain of amino acids. It has 2 cysteine residues. They can come together to form that bond. And this is called an intrastrand bridge. So ultimately, if you have one chain, 2 cysteines, they bond together, that forms a very big ring. So an example of that sort of ring is seen in this molecule called vasopressin. So vasopressin is a hormone that regulates tonicity, or the difference in water potential of solutions separated by a cell membrane. As we read this from the N terminus to the C terminus, we see a cysteine residue. We're scanning along. There's another cysteine residue. Those thiols have been oxidized. They bond together. They form a disulfide bond, and then notice what happens. You get a big ring that forms. So they're linked together. That then affects the secondary and tertiary structures of that hormone. So if you do have those disulfide bonds and you're trying to determine primary sequence, they have to be cleaved. And one way of cleaving them is with peroxyformic acid. So HCO3H, that structure is shown here. This has a peroxide bond, so a peroxy bond. What that serves to do is it cleaves the disulfide bond, and then it oxidizes the sulfur, in this case making a sulfonic acid or L-cysteic acid. So the hydroxy or peroxyformic acid, say in vasopressin, it's going to cleave that big ring that's formed by the disulfide bond. Then you can go through your either enzymatic or chemical methods of determining the primary sequence. So here are some questions to consider. How many rings does vasopressin have? So you include the big ring from the disulfide bond, but also the rings that are present in the individual amino acids. So you're given the sequence itself, but when you treat with peroxyformic acid and then trypsin, how many different fragments would be produced from vasopressin? So where is it cleaving? What are the fragments that are formed? In another example, if you first treat with peroxyformic acid, then chemotrypsin, what fragments are then formed from that? So you'll notice that these will be different fragments because the chemotrypsin's acting on different amino acids. So in wrapping this up, what is the algorithm for determining primary structure? So we know that the individual amino acids, they can bond together to form peptide bonds. You keep bonding them, you get this sequence. How do we determine that sequence? The first thing we would want to do is cleave any disulfide linkages to break cis-cis bridges. The next thing we want to do is site-selective enzymatic cleavage using trypsin, which is selective for lysine and arginine, chemotrypsin, which is selective for phenylalanine, tryptophan and tyrosine, carboxypeptidase, that's selective for the C-terminal amino acid residue one at a time. Then we can do our chemical methods with N-terminal analysis, either with the Edmond degradation that uses the phenyl isothiocyanate, or the Sanger using the 1-fluoro-2,4-dinitrobenzene reagent, and then we can also use digestion. As well in there is the cyanogen bromide that's selective for methionine. So all of these things help cleave a large chain into smaller ones, and then through that fragment analysis we can use some logic to determine what the primary sequence is.